All right, hello everyone. This is going to be our asynchronous work for Monday. We're going to go over assessing how evolution can work, how do we measure whether evolution is happening, and how do we understand the stable changes in populations through something called the Hardy-Weinberg equilibrium. This is generally one of the more difficult ideas for students to understand in AP biology, and so I would take some time to review this lecture and pause where you need to and review the areas that you might want. So this is an example of the timber rattlesnake. The timber rattlesnake is the species that actually evolved over time due to the construction of roads. So the issue is that, these roads are very gray. Before, when the timber rattlesnake lived in the area, it was around these the like darker bushy areas, and so it was able to just be fine. But once these roads were built, the timber rattlesnake started to die off at a pretty fast rate. And so the question that scientists wanted to know, and eventually the timber rattlesnake became lighter, the question that scientists wanted to know was, were the roads the reason that these rattlesnakes were evolving? How could they test for that? How could they know what the reason for the rattlesnakes evolving was? And so that's what we are going to look at today. How do we actually know that a species is evolving? And we're going to look at that through a variety of processes. The first thing to note is the definition of three ideas that will appear very often in this lecture that I just wanted to refresh you all on. The first is an allele. An allele is one version of a gene. Basically, it's your genes often have, they code for different traits, but an allele will give you a certain version of them. For example, in this example of a flower, you can see that the capital B codes for purple, and the lowercase b codes for white. And so the allele just refers to the singular individual gene that codes for it. So this could either be the capital B or the lowercase b. That's an allele. And then you have phenotype. Phenotype is the physical appearance of a trait. And so in this case, the phenotype of both of these flowers is purple. They're both purple. And the phenotype of this flower is white. Obviously, their phenotypes are the physical appearance of the trait. The genotype, however, is the combination of alleles, or the genetics of the trait, or of the organism. So what I mean by that is, here you can see that the genotype is capital B, capital B, capital B, capital B, and you still get a purple flower. Here you see capital B, lowercase b, and you get a purple flower. And this is, in biology, the idea of dominance. So when a gene is dominant, then the existence of one capital allele will automatically turn the flower that color. And so in this case, the homozygous dominant is when there's two capital, and you end up with a purple flower. And so while these two flowers have the same phenotype, they have different genotypes. That's an important idea, so I'll say that again. While these flowers have the same phenotype, they have different genotypes. They look the same, but their genetics are different. And so that's actually going to come into play quite a lot in the next thing that we go over, so it's important to understand that idea. So we're going to actually be studying something called allele frequency. The general idea is that we can actually measure evolution by looking at changes in allele frequency in a population. This is what we call microevolution. Microevolution is the change in allele frequency in a population, and it will show us whether the population is evolving. Essentially, if the allele frequency is stable, then the population is not evolving. The population will not be evolving. However, if the allele frequency is changing, then the population may be evolving. It's possible that the population is evolving. So we are looking for changing allele frequencies when thinking about a population. And again, the allele is the version of the gene. So we're looking for a changing in frequency of the amount of times that either capital B appears or lowercase b appears in a population. So we're going to actually look at this example here of the frogs. And you can see that there's three different colored frogs in this population. So the green frogs, their genotypes are capital C, capital G, for CG. The purple frogs are CGCR, and the red frogs are CRCR. This is maybe a little bit complicated, but essentially the idea is that G here is dominant, R is recessive, but we can differentiate the heterozygous ones. So, and like in the case of the flowers, these would have been the same, but now that they're different, we can actually look at the difference between the two. So what we're going to do is literally, instead of having their color, we're going to write down their genes, what genes each frog has. So we know that the green frogs, they're going to be CGCG. So every time we see a green frog, we're going to put CGCG. Like that, CGCG. And now we come across the instance of a red frog. And so this red frog, however, in this case is going to have the CG, but also it's going to have a CR because it's got a recessive gene in it. So we can look at the gene pool for this red frog. We can say that's, oh, sorry, the red frog is CRCR, my bad. But CRCR here for the red frog. And then we can keep going. We can sort of label every single frog in the population. And so now we have a list of every single allele in the population. And that's really important. We have now a list of every single allele in the population. So now what are we going to do with this information now that we have all the alleles? Well, we're going to try to figure out how many CG is in the population versus how many CR is in the population. So let's look at it here. We can literally just count them, or we can do it a little bit easier math. So I can tell you that there's, let's see, 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15, 16, 17, 18, 19, 20, 21, 22, 23, 24, 25, 26 CGs. So we have 26 CG. And I know that there's 15 frogs, so there's 30 alleles in the population, so there's going to be 4 CR. And we can do a little bit of math. We want to know the frequency, the percentage of CG in the population. So we can actually do this by creating an equation. We know that there's 30 total alleles, right? So we know the denominator here needs to be 30. And we know that there's 26 CG. And so we can do 26 divided by 30. And then here we sort of know that what's remainder is 4 over 30. And here we can find the answers there. So to determine the frequency, whenever I say the word frequency, we're looking for the percentage of the population. So here we can see that the frequency is 0.87 and then 0.13 for the number for CG and for CR. So this is the frequency of the appearance of this population or appearance of these alleles in our population. That's going to be important for figuring out Hardy-Weinberg equilibrium a little bit later. The issue becomes that in a real population, we actually can't track the genotypes of every individual in the population over many generations to see if it's evolving. Like, it's really difficult to figure that out. However, because the genotypes, again, they're hidden, right? They're the things that are inside of our bodies, so we can't actually figure out, like, in every single issue whether we're going to see those particular genotypes. However, we can figure out what the genotype would be for the population that's not evolving and compare it to the population that is evolving. And so we can kind of look at it that way. And basically, this is a blue-footed booby, and we're going to kind of look at these blue-footed boobies in order to figure out some ideas about Hardy-Weinberg equilibrium. The idea of Hardy-Weinberg equilibrium is essentially just to show that the population is not evolving. If a population is in Hardy-Weinberg equilibrium, it's not evolving. That's what we're going to connect here. Hardy-Weinberg equilibrium is a population not evolving. Which means, when we say not evolving, we mean that allele frequencies are not changing. And so when we look at our population, the frequencies that we saw of those alleles should stay the same. Essentially, if our population of frogs was not evolving, what we would see is 0.87 every single time. We would not see this population change from this frequency over many generations. Allele frequency is not changing. And there are 5 conditions in order to meet Hardy-Weinberg equilibrium. There's no natural selection, there's no mutations, there's random mating, there's an extremely large population, and there's no gene flow. These are all of the requirements for Hardy-Weinberg equilibrium in order for that to be true. So we need to meet all 5 of these conditions. And you may be asking, isn't that really unlikely? And yeah, that is super unlikely because most of the time populations are evolving. All of these things need to be true in order for the population not to be evolving, and so it's very rare that they meet all of these circumstances. Okay, so before we get to the equation, we're going to kind of look at a few different things. First is the allele frequency of the dominant allele. We're going to represent this through this idea called P. We're going to use P to represent the frequency of the dominant allele. And so here, I'm just going to calculate how many dominant alleles do we see. And we see 1, 2, 3. We see 3 of them. And I know that there's 5 total blue-footed boobies, and so there's 10 alleles total in the population, so we know this is 3 out of 10, or 0.3. So P in this instance is going to equal 0.3. We have another idea of Q, which is the frequency of the recessive allele. And so the recessive allele, so we know this is 0.3. The recessive allele is going to be the lowercase n. So we know that there's 1, 2, 3, 4, 5, 6, 7. We know there's 7 of those, 7 over 10, and that's going to give you 0.7. 0.7 is Q, the allele frequency of the recessive alleles. The key issue to know is that P plus Q need to equal 1, and that sort of makes sense when we think about the whole population. The frequency of the dominant allele and the frequency of the recessive allele combined will always equal 100%, or P plus Q equals 1. Therefore, P plus Q equals 1. So this is the Hardy-Weinberg equation. We have the dominant allele frequency is P, the recessive allele frequency is Q, and that equals 1. But now we're going to kind of look at another issue of what happens when we're just considering the genotypes together. So here, we're looking at the frequency of NN, the 2 capital N's together. We know that that's 0.2 because that's 1 out of 5. We only see one organism that's capital N, capital N. And so we can see that that's 1 out of 5, or 0.2. And then capital N, lowercase n, again, is 1 out of 5, or 0.2. And then finally, we have lowercase n, which is 3 out of 5. 3 out of 5. Oh, that was really bad handwriting, but that's 0.6. So we can figure out the frequencies of the genotypes as well. And interestingly, all of those added together will also equal 1. And so now we can do some algebra, and we can consider the Hardy-Weinberg equation. We already know P plus Q equals 1, the dominant allele frequency and the recessive allele frequency. We have an additional layer now of P squared. This is what we're going to use to refer to as the frequency of the homozygous dominant, which is what we saw before when we looked at NN, capital N, capital N, P squared. 2PQ will equal the frequency of heterozygous, capital N, lowercase n. And then Q squared will equal the frequency of homozygous recessive, or lowercase n, lowercase n. And all of these will be equal to 1. You never have to memorize this equation. This will always be provided for you on an equation sheet on the AP test and on anything that we do. But this will be sort of you will have to understand the meanings of each of these variables. And so I just want to put together these variables for you. we'll use this equation in order to understand and figure out whether a population is evolving, and we'll practice that when we get to class. But it's useful to have all this information down so that you understand where this equation is coming from. And yeah, so essentially, we know that this will determine whether a population is evolving or not, because if the numbers don't match this equation, if p squared plus 2pq plus q squared does not equal 1, then the population is evolving because it's not in equilibrium. That's what we are kind of going to figure out. Here, we kind of understood that the population was evolving, but if it doesn't equal 1, then it becomes an issue. So we're going to go over a lot of the practice with the stuff. I know it's very complicated, so we're going to review it in class and then hopefully work on some equations to figure out if some populations are evolving. But we'll stop right there. Similar deal, take your notes on this and submit the notes on Canvas. All right, have a good day.